In this video, we will discuss about the development of this mKRAS vaccine along with ipilimumab and nivolumab drug to activate T cells against the mutant KRAS cells or cells that have this mutant KRAS. And this is at right now at the first stage of the clinical trial. So we will understand their developmental stage or what are the results that they got from their developmental period. So in order to start with their vaccine development, what it is, they take the patients and uh, and the eligibility criteria for the patient is that they should have undergone the tumor resection in the past or in the last 6 months. Then only they are eligible for their vaccine trial. So what it is, they take the different mutant KRAS that is mentioned here, that is G12V, G12A, G12R, G12C, G12D, G13D, and these are the common KRAS mutant that are found in the pancreatic cancer patient. So they started with this mutant, and the idea is that if this peptide of this mutant KRAS injected into the patient, then patient immune cells, that is T cells, will develop antibodies against this peptide, or we can say that it will develop the antibodies against the mutant KRAS cells that contain that specific mutant of the KRAS. And the history of the patients who has undergone this vaccine trial is shown in this panel B, where we can see that most of the patients undergo some kind of treatment, such as this mFluorouracil treatment. The first time the vaccine injected is also shown here in the red diamond, and the other treatment that they went through. So all these patients has already went through the first treatment from which they did some tumor resections. So they are eligible for their vaccine treatment trials. So what they are able to find out that when they inject peptide of different mutant mKRAS along with these drugs, that is ipilimumab and nivolumab, they are able to see increase in the amount of IFN gamma. The IFN gamma is usually released by the activated T cells. So in most of the cases, they are able to see increase in the amount of this IFN gamma, and those results are in line with this mKRAS vaccine and ipilimumab and nivolumab. So since we can see that in most of the cases the signals went up, indicating that there is increase in the activation of the T cells post vaccinations. So what they are able to do in short is that they are able to activate the immune system of the patient who are affected with pancreatic cancers against the pancreatic cancers. So normally what happens is that pancreatic cancers is a solid tumors, so it has this tumor microenvironment that act as a protective layer, and along with that the different glycosylations within the pancreatic cancer cells also provide a glycocalyx which has the compositions of different carbohydrates that also acts as a physical barrier to prevent the cancer epitope to be detected by the immune system. So they are providing these epitopes externally through these different peptides that helps in the activations of the T cells against these mutant KRAS cells that are present within the patient. So in short, they use they externally activate the immune system of the patient against the pancreatic cancer tumor cells, so that it can act against the pancreatic cancer tumor cells by generating antibodies that is specific to mutant KRAS cells. So this panel C just reflect their results of the idea that they can activate the immune system externally instead of making the immune systems interact with the tumor cells through the immune checkpoint blockade therapies. So they have also mentioned that when they use the immune checkpoint blockade therapies, it has given the grade 4 immune adverse effect. So that's why at this point of time they have not included this immune checkpoint blockade in this case. However, later we will see whether they have again included this ICB based on some modifications or not, as their heading of the paper is that it can produce the vaccine from this mRNA peptides along with this dual checkpoint inhibitors. So we will discuss what are the results that are presented in the next figures in the subsequent videos. So I hope you understood to some extent how they are able to activate the immune system of the patient externally against pancreatic cancers. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.